To a cell biologist, a person is really just 50 trillion cells walking around cooperating with one another. Whether you like that view or not, to understand human biology, we have to have some idea how each of those cells works and how they're cooperating with one another. This series of videos is going to focus mainly on the process of how proteins are synthesized and how they're sorted out in this process over here with the endoplasmic reticulum and the Golgi bodies, so that each protein finds its own home and become active in its appropriate compartment. On the last slide, we showed a typical human cell, but of course no such thing exists. For example, macrophages like this are relatively small and they can crawl around and engulf things. Neurons like these guys can be over a meter long. Muscles are made up of groups of cells that have fused together and formed syncytia, so they aren't cells at all. So inside the human body, there are all sorts of different kinds of cells that all have different functions, and they all have different architectures that allow them to perform those functions. Not only does every cell have to have a particular composition so that it can perform its function, but every compartment within the cell also has to have a specific composition of proteins so that it will have the appropriate catalytic capacity and the appropriate structure. So here's three guys over here that all have different fates. One of them, this green guy, is being exported out of the cell. This blue one is sitting in the plasma membrane performing some function where it needs to be sitting at the surface of the cell. And actin over here is performing a structural role bound to this membrane protein so that it can give the cell its structural integrity and perhaps perform other functions. So those are three very different fates for proteins, but all of them are coming from messenger RNAs over here that are being exported out of the nucleus into the cytoplasm where the proteins are being synthesized. So some sort of a sorting process has to go on so that each of those proteins can end up in the appropriate compartment. No matter where they're going to end up, all of these proteins start off as messenger RNAs that are being synthesized, translated into proteins by ribosomes. So the ribosome engages the messenger RNA, and in each catalytic cycle, it adds one more amino acid to a linear chain, ending up with this long string of amino acids that will form the protein. Now, usually we just pretend that that linear sequence of amino acids magically folds itself into its tertiary structure that it needs so that it can perform its function. But while we're thinking about sorting, we have to be a little bit more realistic about this miracle that happens at that step. Because this doesn't always work, and it's the job of the sorting pathway not just to bring things to the appropriate place, but to do some sort of quality control to see whether those proteins are any good. This is going to become an enormous problem if we have mutations in the genes that encode the proteins, which we'll talk about in the next video in this series. But I just wanted you to think a little bit about how hard it is for this linear sequence of amino acids to bring to adopt its appropriate secondary structure. The only principles I want you to have in mind are that hydrophobic amino acids are going to, during the folding process, hydrophobic amino acids are going to form the core of a globular domain. That's going to drive this structure to form. And that's going to leave the hydrophilic residues out at the surface of the protein so that the whole thing can be soluble. Now that's a nice simple rule, and we'll look at mutations that violate that rule and therefore cause proteins to be difficult to fold. But even that rule is too simplistic to look at real proteins. So I've got an example down here at the bottom where this guy, histone H4, those are the amino acids in its linear structure, and it has folded up into a secondary structure, 3 alpha helices that are shown as ribbons here. But ultimately, H4 has to work in a context with 7 other proteins down here in a histone octamer. And that octamer really only has any function when it forms a nucleosome with DNA wrapped around it. So if now you look at H4 and you say, okay, this linear sequence of amino acids has to drive a hydrophobic core so that it can make this structure, but it has to fit together with all these other proteins ultimately, and those guys can only be assembled properly when they've got DNA wrapped around them. So this makes this folding problem look a little bit more difficult, I think, than you might have imagined it to be. How do we get this guy over to where these guys are and the DNA is all intact and folded up and ready to work? So we're not going to have any answer to that question. This is something we really don't even understand very well right now. But it does sort of pose the problem. How do you fold a protein properly, and how do you check to see if that's happening? And that'll be part of the subject of this sorting process. If a protein doesn't fold properly and make it to the native folded state over here, it doesn't just make a useless polypeptide. Some of the folding intermediates can aggregate with one another after a nucleation event like this and form other alternative structures like amyloid. If we get enough of this garbage filling up a cell, we can cause all sorts of trouble within the cell. This is thought to be one of the initiating events for diseases like Alzheimer's, is to form these amyloids in neuronal cells that then are difficult to resolve. So folding is a significant problem. Natively folded proteins aren't much more stable than the folding intermediates, so it doesn't take much to push things over the edge to cause a real problem. Okay, but back to the sorting issue itself. Here's an outline of the different fates that a protein might have. We'll be talking mostly about this particular one where we'll look at the endoplasmic reticulum as a sorting station for a large number of proteins. Mitochondria and plastids and peroxisomes have similar systems. We're not going to talk about them. The only one I want to cover in this particular part of this video is this nuclear localization pathway because it's quite distinct from the others. Now, we talked about how different proteins have to get to different places in order for them to be recognized as going to those compartments. They have to have signal sequences on them. So these particular signal sequences are not unique. We can't recognize them necessarily, but we do know that if we put a particular protein sequence while it's being synthesized, or in this particular case, after it's been synthesized, we can add this signal sequence to a protein, and that is a signal that says, take me to the nucleus. We know this is true because we can actually take that piece of protein and either have it encoded in the gene or chemically modified and added to a protein. So here's an example where pyruvate kinase, we're looking at cells here, so this is immunofluorescence where we have made this protein glow green so that we can see it inside of living cells. And the pyruvate kinase, normally when we do that, is filling up the whole cell here. It's sort of excluded from the nucleus, but it's found all over the cell, and this makes sense because this is a cytoplasmic protein that has a function in the cytoplasm. But if we attach this piece of protein from a normal nuclear protein, in this case the SV40T antigen, then all of a sudden the pyruvate kinase thinks that it's supposed to be in the nucleus, and it goes to the nucleus and is not found in the cytoplasm. So we think we understand something about this process of how we can tag a protein so that it will go into the nucleus. The thing that then has to recognize that is what we'll talk about next. So here's the nuclear localization signal we talked about, and the nuclear import process involves these pores that are sitting in the nuclear membrane. So here's a nuclear membrane. It's going around the DNA on the inside here, the inside of the nucleus, and the cytoplasm out here. A protein like that pyruvate kinase that was synthesized out here in the cytoplasm that has a tag on it can be recognized by this complex. If the protein is small enough, it can actually just diffuse through these pores. They're actually big gates that allow things to flow in and out of the nucleus. Small molecules can flow by themselves, but a protein like pyruvate kinase usually is too big to get through that hole, and it has to be recognized here by some recognition machinery that then says, okay, you're the sort of thing that ought to be in the nucleus. I will allow you through. This can all happen while the protein has been synthesized and folded into its normal tertiary structure. So this large gate allows big things to pass through. And this is convenient because there's a lot of stuff that has to shuttle in and out of a nucleus, and we don't want to have to make it go through a membrane every time, and we don't want to have to unfold it while we're doing that. So lots of stuff has to come in. So for example, something that has to go in, pick up a messenger RNA, and then bring that messenger RNA back out, now it can shuttle back in to the nucleus, pick up another messenger RNA. So the nuclear pore is a sort of specific case of sorting where the recognition occurs at the pore and large folded proteins can get in and out of the nucleus. So that gives you some idea of what the problems are. We've got a single source of messenger RNAs from the nucleus. Those messenger RNAs all have to be translated into proteins by ribosomes, but then different things have to happen to those proteins. Some of them have to get outside of the cell, some of them have to get into membranes, some of them have to get back into the nucleus. We saw how parts of the polypeptide sequence can say, bring me into or out of the nucleus, but how do we get things into this sorting pathway through the endoplasmic reticulum and the Golgi apparatus? That will be the subject of the next part of this video.